Welcome. Here we are. I'm here with Leonie,
Hi guys.
and we're going to talk a little bit about the slides that we looked at in the first laboratory with some tips and tricks for identifying them. So let's start right off. What are we looking at here?
This is the squamous epithelial tissue, and what I look for is it's a flat layer that looks scale-like to me. It could or looks like paving stones or cookies on a sheet.
Mm-hmm. Yeah, how come this doesn't look like, where's the free space on this? How come you can't see anything?
This is a hole mount, so it's taken straight from the top, so you wouldn't see the free
Oh,
space on this
okay,
slide.
yeah, okay, so we're looking straight down on this. Yeah, so each of these, and you notice how it's called, we call it squamous tissue, so you can see each of these individual cells, but yeah, not the, not the any kind of layering to them. So that's why you're kind of missing a layer part. Yeah, okay, so whole mounts we're going to rarely see those. Okay, simple squamous epithelium. This just looks like one big mass of tissue. Where's the free space here?
So the dark stained nuclei actually fill up most of the entire cell.
Mm-hmm.
And this is a slide from a kidney, so this free space is, I guess, around the cell.
Yeah, so the layer that we care about is this kind of, the free space kind of goes around this little island of tissue right here. So the white space is there deep to that. And yeah, I agree. You can see that the cell's nuclei are pretty large, and they make a lot of the area that we're concerned with. Yeah, so the kidney tissue is kind of its own kind of animal. Okay, but again, one thin layer, and what's surrounding it, as it turns out, are simple cuboidal epithelium. And here's another view of it elsewhere in the kidney. So what are like the distinguishing characteristics here?
For me, I look for cells that have their nuclei taking up almost the entire cell, and I think it's easy to distinguish because it's just one layer of roughly cube-shaped
Yeah,
cells.
I buy that. So what is this a slide of? I mean, what's actually getting cut in this field of view? Do you know?
This is also a kidney slide.
Yeah, but I mean, what structures are being cut so that you have like these little oval shaped or round shape kind of spaces? Well, that's okay. These are tubes, right? A real common way that we see simple cuboidal epithelium occurring is as basically making up the walls of tubes, and the free space here, the open space, is basically the lumen inside a tube. Some of them look round because we're cutting them right in cross section. Some of them more oval because we're hitting them in obliquely.
Okay.
All right. Now, simple columnar epithelium. Again, where's the free space here?
The white space that's outlined around or inside of the blurred line that you can see are the microvilli.
Yes. Okay. Yeah. So, how do we tell? So we have some basic characteristics here. We can see that there's a little space here in the center. The cells are tall, and the nuclei tend to. It does seem to be like a zone of nuclei, where they're not all kind of randomly jumbled through the cells. So most of the working like absorptive parts of the cells are toward the apical surface. And we're looking here at like almost like this little crack within some villi in the gut. So and again, and this looks pretty nice. Showing the microvilli, like this haze of microvilli, not like a sharp apical surface like we saw in the preceding slide. So here, those would be flat surface cells, whereas the apical surface here, those have those microvilli. This is like nicer because I like the kind of fuchsia as opposed to the regular purple the cytosol.
Yeah, I think it made sense what you said in lab that the microvilli appear as though you took just a Sharpie and draw right over the top of the surface.
What a good example. All right, so let's see. Okay, ciliated simple columnar tissue. All right, so here, I mean, I know part of my rap is that transitional pseudostratified epithelia looks like it's got tons of nuclei. It looks like there are a lot of nuclei here, but I don't see this. It doesn't look like there are like thousands, so you can still sort of see how the nuclei, they're all basically lined up like one cell.
Yeah, they look
width
like they're in the same general area of the cell.
Now compare what the cilia look like. Actually, this is a really nice picture of these cilia. They look like little hairs.
Yeah, they're definitely more distinct in this picture than in the microvilli in the previous, which is just hazy.
Yep. So they're just all blur because they're so thin. But these guys actually have not only some space between them, but they're a little bit more, a little bit tighter. Yeah. All right. Now, here, this is a slide from the handout, which actually now when we blow it up looks a little bit blurry. But in the case of the pseudostratified ciliate epithelia, again, here, obviously one nucleus per cell. Here, it takes a while, but you figure out there's basically one per cell. Here, it just looks like there are tons of nuclei. This is a slightly different magnification, though. And yeah, the cilia look a little bumpy here, but yeah, they're also reasonably distinct. All right, cool. So now, free space up to the top again. So talk to me about this kind of epithelium. How do you identify it?
So this is stratified squamous epithelia, and a characteristic that I look for is that it's usually resting on top of layers of cuboidal cells, which I think you can clearly see here. And it's the same flat scale-like structure that you look for. There's just many layers of it.
Right. So, as always, we name the tissue type based on what it looks like at the apical face. But yeah, they start off being kind of bunched together. Do you think this is the epidermis? Usually when we look at the epidermis, I mean, it's nice because you got, like, there's sort of a color change here at, like, the base of the tissue, but you don't see how, like, there's still, like, a lot of open space here. Like, the cells aren't particularly packed together. If this were the stratum corneum of the skin, those guys would be all really flat, and also we'll see when we look at the skin later that it's really, they really pick up the stain a lot too. So it almost has that really kind of dark fuchsia kind of color to it. This is probably the esophagus. Another view of it here. Similar here, we do have a little bit more of the of the top here. We see it flattens out again. It does pick up the stain. Like they get a little bit thinner, but again, this isn't going to be skin, I don't think. All right. Now, transitional epithelium. We have two varieties here, and these are slides that everybody has from the handout. So there's like the relaxed appearance, and then the more stretched appearance. So, how do we differentiate these?
These are actually the two cell types that I find the hardest to differentiate. So I don't have the greatest tips on identifying these ones.
Okay, well, one thing I would do is, there are a few places along here, usually where it almost looks like there are these little bubbles that are up at the free space. Now, but we don't see that same kind of bubbly appearance
on the stretch.
Yeah. Oh, these little things here, like kind of along the surface. That's just where the blade cut the tissue and just kind of like scraped off it. So that's a little bit deceptive, but I think that relaxed transitional epithelium is more distinctive because of what look like almost like kind of roundish bubbles, like at the free space.
Okay.
Because remember, with transitional epithelium, the cell layers kind of slide past each other. So here, I think this is pretty like that. That makes the relaxed transitional epithelium relatively distinctive. But when you look at the stretched kind, it's hard to see cell outlines. It's hard to see layering. It's hard, like it is tricky to be able to do that.
Yeah.
One. That's partly an issue with this particular photograph, but that's one thing that's kind of typically going to be true. So for my money, for things that we can do easily in lab, I'm much more likely to ask for relaxed transitional epithelium for the reason that there are distinctive characteristics about it.
Structures.
Yeah. Because this, the other one, it would be hard to say. I mean, I would be hard put to figure out what the shapes of those cells is. So, I'm not gonna ask the impossible on an exam. So, if it makes you feel any better, if you're confused about this, focus on relaxed transitional epithelium because I think that one is more distinctive. The stretched one, hell, I might even ask like a leading question, like this tissue was found lining the ureters or lining the urinary bladder, or something like that. So
it accommodates stretching and expansion.
Yeah, so that like urinary system association, that's the only place where we see transitional epithelium in the body. Ooh, this is pretty. How come our slides don't look like this?
The staining on this slide's much better.
Yeah, well, it's probably not 50 years old. That's probably the problem. All right, so, yeah, talk to me about this.
So you can see the dark staining nuclei, which are the fibroblasts of the tissue.
The fibroblast nuclei, right?
And then you can see the thick pink lines, and those are the collagen fibers. And the very thin dark purple lines, those are the elastic fibers.
Right.
And I kind of think that this slide reminds me of a dryer sheet with all of like the fibers in it. That's what helps me
Yeah, I like
identify
that.
this.
Yeah, that's good. That's good. So, I mean, you could, regular, like elastic connective tissue, we'll see a little bit later. So, areolar connective tissue, yeah, what's really distinctive about it are like all those voids where, and it's funny how you can see the fibroblast nuclei but not the outline of the fibroblasts. I don't know why that is. I think that it probably has to do with the kind of stain they use, but that's a very kind of common. That's a very kind of common appearance to these guys. What the hell am I looking at?
Adipose tissue.
It doesn't look like anything.
It's looks like soap bubbles, I think, and we kind of talked about that in class, and that's how I use to identify it. And the white spaces that you see are where they're storing the lipids.
Yeah, big old fat vacuole in the middle. Yeah, the nuclei aren't super apparent in this one, but they ordinarily, and actually, based on how it really, you can see like the outlines here, and these sort of kind of overlap. This looks like, again, it looks like the blade kind of cut and smeared some stuff around. So this isn't quite as, I think our stuff might be even a little bit better for a change. But yeah, when you look around here, like, so let's say if you look at this cell, what you can, this is probably not a capillary. I think that's probably the nucleus smushed off to the side.
Okay.
This is probably a capillary over here. Oh, this doesn't look like anything.
Dense irregular connective tissue. These they don't have a preferred orientation, which is why it has the wavy appearance.
When you say they, what do you mean?
Oh, sorry, the dense irregular.
No, but I mean, what's the orientation? The orientation of what?
Oh, the fibers their grain isn't a specific orientation, so they lack that. And so you're just seeing the thick collagen fibers and the dark staining fibroblasts, the nuclei of the fibroblasts again.
Mm-hmm. Okay, so if we compare that to here.
So these fibers do have a preferred orientation. So you'll always see them going one specific way compared to the irregular connective tissue, which is all over the place.
Yeah, so like if we can, so it's mainly collagen fibers looking at here. So regular tissue would look like this, where there's clearly like a, northeast to southwest kind of orientation here. Here they're all over the place. And just to go back a little bit, this is when they're not packed together. So this would be why we call this loose connective tissue with all the voids. Whereas this is dense because there aren't as many voids, but it's all the same kinds of collagen fibers that we're looking at. And this one it's hazy on the screen, I guess. But this one you can almost sort of get the impression of the collagen fibers being made up of individual like proteins that are kind of belted together. Yeah. Interesting. All right, elastic connective tissue.
So elastic connective tissue, the elastic fibers are darkly stained, and the example given in class, I think, is really good that it looks just like layers of lasagna.
Yeah, I buy that. These aren't as apparent, though, are they? I mean, here, like, I can see, like, kind of near, like, right around here, I can see it okay. Yeah, and so like once you clue in on that wavy appearance, basically going left to right, then it becomes more clear. Yeah, I think our stuff actually looks better because it picks up the stain a little bit better. Yeah, but that's great. So sort of this little interspersed collagen with that wavy elastic.
And I think they also have more or less a pattern, like a specific kind of direction they go in, more or less.
I agree with that, but we never call that like regular elastic connective tissue. And I suppose we could. I don't know if it's because the dense regular connective tissue, it's like linear, whereas we're looking at, for most kinds of elastic connective tissue, lives like around the aorta, like big blood vessels that are pulsatile. We're looking at like the bottom part of a section, like the wall of a tube. So here it looks like they're going straight left to right, but in real life they're actually going circumferentially.
Around.
So that might be why they don't do it. But that's a weird one. Oh, one thing I want to ask here: what kind of like tissue, what kind of organs would you expect to find something like this in a dense regular connective tissue, as opposed to the irregular? Well, all right. Well, I mean, again, if there's always these guys, remember, collagen fibers are great in tension for pulling. So what you hear is when, like, if you have a tendon or a ligament where you're always going to be getting, like, think like your calcaneal tendon, your Achilles tendon, you're always going to get stretch in the same direction.
Tension.
So tension in the same direction. Whereas here, this is great in the reticular layer of the dermis, where you've got lots of different, you don't know, there's no preferred orientation because it's always getting pulled and pulled and stretched in different
ways.
directions. All right, on to the cartilages. This is like your base model of cartilage. So walk me through hyaline cartilage.
So the white spaces that you see are where it's the lacunae where the chondrocytes live, and normally below you would see pseudostratified columnar cells.
If we're looking at the trachea, yes, but if this were bone, then if this were like part of the growth plate of a bone, then we wouldn't see that. So this is a typical thing where we see like massive extracellular matrix without a lot of fibers, without much space. So this is just those proteoglycans and hyaluronic acid and stuff like that that's in the hyaline cartilage. Okay, so this is the base model. So when you, if we were going to turn this into fibrocartilage, what we would get is you'd see more of, you see a little bit tighter. I'm going to have to edit this part. I'll have to note to self to edit this. We would see, if we were looking at fibrocartilage, then we would we'll have more apparent fibers, and it's hard to tell fibrocartilage from elastic cartilage sometimes until you can compare the two directly. So elastic fibers are really distinct because they're so dark, and fibrocartilage is sort of this in-betweener where there are a lot more fibers, but you can still see lacunae, and it's that same basic kind of ground substance around the cells. Yeah, all right, good. All right, and bone looks like one thing in the world.
Yeah. So the concentric rings that you see, those are the lamellae, and they're around a central canal where you'd have your blood vessel. And the osteocytes, to me, on a slide, they just look like fuzzy dark spots that you can see within the rings.
They're fuzzy, actually, when we're going to see this when we talk about bone as a tissue. They're fuzzy because there are these little tendrils almost that extend from them. So it looks like it's blurry, but in real life there are little cracks. Those things are called canaliculi, so that allows each individual osteocyte to be connected to other osteocytes and also to have access to the central canal so they can get nutrition and stuff. Skeletal muscle, nice one.
So for the skeletal muscle, they're multinucleate and they have distinct striations, which you can see here, and the nuclei are often along the periphery of the cell.
Why are they multinucleate? Don't cells usually just have one nucleus? Good question. It's because skeletal muscle, we usually call these skeletal muscle fibers for the reason that they are fusions of multiple cells. So the cell's nuclei, so these cells, individual muscle fibers can be really long, they can go the whole length of your thigh muscles, So we're looking at, a foot for some people. So in this case, we're looking at actual fusions of smaller cells that kind of organize so that you can have these really distinctive railroad track kind of appearances, barrel-shaped cells, fibers. I'll get inattentive about fibers versus cells later on. All right. Ooh, this is a nice one.
So for cardiac muscle, they're short branch striated cells that are more spaced out than you see in the skeletal muscle. And in this slide, you can see the intercalated discs between, which are the dark vertical bands that border they're borders between the individual cells.
Yeah, this is much clearer than what we usually have. Yeah, so usually the intercalated discs look like darker striations, and you're correct in that they mark the borders from one cell to another, and that's where there are gap junctions to send along the contractile impulse. We'll talk about how the heart works in the spring. Smooth muscle doesn't look like anything.
So the smooth muscle have single nuclei compared to the multinucleate cells we saw before, and they are arranged in perpendicular sheets, and some of the cells would be elongate versus circular. And one important thing to note is that this type of muscle isn't striated compared to the skeletal and the cardiac.
Right, so this would be an individual smooth muscle cell. So they're kind of tapered in their appearance, and no striations, because as it turns out they do have contractile proteins in them, but they're just not arranged in the super regular fashion like you see in cardiac or skeletal muscle. Ooh, that's pretty. This looks like a science fiction thing or Cthulhu or something.
So for nervous tissue, there's the dark staining nuclei that you see around, which are the nuclei of the supporting cells, and for the cell body, which contains the nucleus and the cytosol, and then there's the axons that are seen extending from the cell body itself.
I would be hard pressed to tell you which one of these is the axon. When you have a multipolar neuron like this, where it looks like this thing almost looks like an octopus in the sense that it's sending tendrils out in all directions. I mean, maybe I could be convinced that this one down here is the axon, but without seeing more of it, it's hard to tell. But yeah, you can at least tell the difference between the cell bodies
and the supporting nuclei.
Yep, the glial cells nuclei, as well as at least the processes. So I wouldn't be picky about, especially without a pointer in the scope. I wouldn't be picky about what's a dendrite versus an axon. Okay, good. So some skin. This is labeled, but that's okay. Now, all right, good. So let's walk me through this.
So for this slide shows the layers of the skin. So you start on the apical layer, which is the stratum corneum, and that's about 20 to 30 cell layers thick. And this slide actually shows the stratum lucidum, which is only seen in thick skin. It's a thin layer. And then there's the stratum granulosum, which are a few cell layers, and those are more darkly stained than the others. And the stratum spinosum, for me, I just recognize that as being the layer that's right above the basal layer. And then the stratum basal is lining the dermis.
Yeah, I buy that. It's interesting. Here, stratum lucidum is usually light in coloration. This is a little bit unusual in that you get kind of a darker stripe here, but I buy that for being the stratum lucidum. It's in the right place. Here, what the stratum granulosum, this is where like your lamellated bodies or like your little the little vesicles of prekeratin, that's what's picking up the stain there. That's why it has that really dark appearance. But I also like this sort of process of elimination. If you know that this is the stratum granulosum and you know that the very bottom layer here where the color change is where the stratum basal is, and then we know the stratum basal is only like 1 or 2 cell layers thick, the spacing between them has to be that stratum spinosum. So like it's okay to kind of back your way into figuring out what you're looking at. Like that's a really common approach, and I think a good one for kind of getting used to how to work with slides. So, what parts of the dermis can you see here? I mean, what do we call this part that kind of pokes up into, like kind of it pokes up into the epidermis?
So the papillary layer that shows the dermal ridges.
Yep. So there's papillary layer, and actually, just deep to that, like I'd say even from here to the bottom of the slide, that would be where the reticular layer starts. And you can see that pinkish, like those are all collagen fibers that are dense, irregular fibrous connective tissue.
Yeah, you can kind of see that there's no orientation down there. It's kind of going every which way.
Yep, more like swirly. Good. Nothing to do with thick skin. Interesting. So, what's consistent? Here it looks like the stratum lucidum is like this pink color. That's weird. So weird. But again, this is blurry, but even comparing it to what we saw in this slide, you can see that the stratum basal, there's a real color change between the papillary layer of the dermis and where the epidermis starts. And then again, the stratum granulosum also picks up, those grains really pick up the dark stain. But here, it's interesting how you really, once you get into the stratum corneum there, those cells are really just flattened out. It's hard to even see the border. I mean, blurry, but still, you can really hard to see the borders. Oh, okay. Great. So what am I looking at here?
So this is a slide of a hair follicle, and you can see the sebaceous glands wrapping around the follicle itself, kind of in that donut shape that we talked about in class.
Yep. You promised sweat glands. Where are there sweat glands here?
Are they down at the
you can
bottom?
See them kind of cut here a little bit. It's actually a little bit funny. There's some over here in this corner. Most of the glands you're just seeing are sebaceous glands, and sebaceous glands have that. It almost looks like stratified. I mean, squamous tissue again. Yeah, it's like got that kind of
scale-like. They're flat.
But they're probably in real life they're probably more spherical. They're probably more like the adipose tissue soap bubble than they are like the cookie sheet kind of thing. But down here in particular, where you can see that there are little cuboidal cells that are surrounding a lumen. That's pretty clearly some kind of eccrine sweat gland. This I could be convinced is one as well, but it's just a kind of a funny cut. Okay, so we've been through most of the main kinds of slides, and I think that this none of these slides is going to be exactly the same, either as, what we've showed here or, the photos that appear in the lab handout. But the thing to clue in on that I think we did is talking about, like, how do you tell the difference between them? And I think as with practice, everybody gets good at being able to, reason their way to what it is that they're looking at if you kind of sort of internalize what the basic rules are. All right. Well, say good luck.
Good luck, guys.
Very good. Was that so bad?
No, I just get really